Good morning, everyone. I'm Gregorio Sánchez, and I am currently involved, although I work as a postdoctoral researcher in the Natural History Museum in Madrid. I'm currently involved in collaborations with Polly and Ivan here at the EBD. So I appreciate the opportunity of participating in these EBD talks at the capture-mark. And I want to talk to you about what we call integrative demography. And we use that name because we combine different lines of evidence, mainly genetic analysis and capture-mark-recapture data, to solve some questions regarding the dynamics of populations. So this is our work team is led by Íñigo Martínez Solano from the Natural History Museum in Madrid. And this is Jorge. He's a former PhD student who set up the beginning of this research project with amphibian demography, along with Íñigo and myself 10 years ago. So the main point here, why demography? We study demography because we think that if we want to get a deeper understanding of what are the processes that drive to the patterns of diversity, of biodiversity distribution that we observe today, biodiversity understood from the genetic diversity level to the communities, we really have to integrate different scales and different levels, spatial and temporal scales. And from these different levels, different scales, we think that the local scale, which is precisely demography and the natural history of a species, is often disregarded and we think poorly understood and developed. So let me give you an example to illustrate this point, and this example is from our own research on the group. So for example, these are two Mediterranean species, the sharp-ribbed newt Pleurodeles walt and the spade-foot toad Pelobates cultripes. And for these two species, phylogeographic studies have recently been developed. And what the data show is that both species show a gradient of genetic diversity depletion towards northern latitudes across their range of distribution. So this is consistent in both species, and interestingly, this pattern of genetic diversity decrease with latitude is strongly correlated with the stability in climatic favorability through the Pleistocenic glaciations. So red colors in the left figure represent higher genetic diversity. Red colors in the right figure represent areas that have remained stable and favorable for each species. In this case, both figures are for Pleurodeles walt, but the pattern for Pelobates is the same. So these are areas that have remained stable and favorable through time, through the Pleistocenic glaciations. So this information is obtained from species distribution modeling, which is the area of expertise of Marcia Barbosa, our collaborator from the Évora University. And our interpretation of the strong correlation of the genetic diversity patterns observed today with the stability in climatic favorability for the species, our interpretation for that pattern is that southern areas in the Iberian Peninsula really act as a refugium where the genetic reservoir of the species was kept during unfavorable times, during the ice ages. And then recolonization of newly available areas, when the ice sheets retreated, started from this refuge. And the fingerprint of this recent colonization is the genetic diversity depletion that we observe towards the north. So, if large areas of the Iberian Peninsula became unsuitable for Mediterranean species during the Ice Age, what happened? Is it possible that more Atlantic cold-tolerant species that nowadays inhabit the very same places that we can see in sympathy with those Mediterranean counterparts, is it possible that these Atlantic species show fingerprints of a different Pleistocenic history or historical demography? Well, to address this question, this year we published this work in which we explored the phylogeography of the Iberian tree frog across its range, and we detected shallow but significantly resolved genetic structure through the range of distribution. And interestingly, we found that the favorability through the Pleistocene in this species didn't show very strong differences between different parts of the current range of distribution. So apparently, more or less, the current range of distribution correspond to areas that remained favorably through the Pleistocene, more or less. And furthermore, the genetic structure or the genetic diversity distribution of this species is not concentrating mainly in some putative refuge, as in the Mediterranean species, but is non-homogeneously widespread throughout the current range of distribution. So this is a difference of this species with respect to the Mediterranean species which with it currently cohabits. And our interpretation of this difference is that this species didn't seem to be affected by the ice ages in the same way as the Mediterranean species. So maybe this species didn't suffer such extinction and recolonization processes throughout the climatic oscillations in the fluctuations in the Pleistocene, but instead maybe it was able to accommodate those differences in climatic favorability or in climatic in climate in terms of its cold tolerance, its thermal features or traits, and its dispersal capacity, allowing it to trace the differences in climate, the arrival of the ice sheets and the retreat of the ice sheets by changing and moving its range of distribution through time. Moreover, if we are assuming that this is the explanation for this pattern, we need that this species showed relatively large effective sizes to provide migrants for these accommodations for the range expansions to occur. So we are making a strong assumption of the life history traits and the natural history of this species in order to explain the difference that we observe with Mediterranean species. So our point here is that we need a deeper understanding of a solid background on the natural history and demography of the species to be able to make such strong assumptions and to test our historical inferences with ecological data. So if we can do that, we will obtain a deeper understanding of these processes that led to current patterns that we observe today. So, okay, so demography is important, okay. So if it is so important, why do you think we know so little about the demography of a species? Well, of course, I don't have to tell you that population monitoring in every taxa, in every species, is challenging. And amphibians, of course, are not an exception to this. And why is so? Because if we read the literature, available studies about long-term demographic studies with amphibians show us that amphibian populations show wide fluctuations in abundance. And these are natural fluctuations that are not often associated with population trends. They are just natural fluctuations. So it is difficult to know what is happening to a population because it naturally fluctuates a lot. And also amphibians show a very secretive way of life. They are difficult to observe, mostly throughout the year. So what do we need? What do we need? We need protocols to standardize demographic studies in amphibians, but in all the other taxa. We need optimized protocols. And we have our bets in this integrative proposal which combines different lines of evidence, but mainly genetic and capture-mark-recapture, in order to estimate the relevant parameters. And which are the relevant demographic parameters? Well, there are, of course, a lot, and they are classic in the literature, but I will focus on two of them today in this talk. And the first will be the effective to census population size ratios, and on the other hand, the connectivity between different populations. Okay? So first, the effective to census size ratio. What is that? So if we just said that amphibian populations show wide fluctuations in abundance, we cannot just count frogs to know what is happening to the population. We do not just need to know how many individuals are there in the population, which is itself quite difficult to estimate, but also we need some idea of how many of them breed in the population, and even more, what is the relative genetic contribution of the offspring to the offspring? So what is the average individual breeding success? And those things are very, very difficult to estimate. That's why we have this lack of information, but today we have the tools to address these questions in integrative frameworks. So we use capture-mark-recapture to estimate the number of adults in the population, and we use genetics to estimate the effective size, which is approximated by the effective number of breeders, NB. So this NA and NB are the only two abbreviations that I will ask you to keep in mind during this talk. So NE is the number of adults in the population, NB is the effective number of breeders, and we will get into detail into this. So NA maybe is the most straightforward, the more classic parameter, and it can be estimated by different statistical methods based on capture-mark-recapture. So to this end, we have been monitoring amphibian populations in central Spain since 2010. Using a passive integrated transponder, so tagging individuals and recording them individually and tracing their capture histories. And we use a special method called the robust design. And this robust design is especially interesting for us studying seasonal breeding species. And why is that? Well, that is why this is because the robust design is based on a nested hierarchical type of analysis which tends to congregate the sampling sessions in short time frames. What is the advantage of that? Well, if we can concentrate our sampling sessions in a short time frame, then we can assume that the migration and the recruitment and the death rate during that short time frame can be neglected. So if that can be neglected, then we are sampling an approximately closed population And that means that we can estimate the size of that population. Then, between those short window time frames, then we can estimate the survival, and then we can estimate the migration. But we can estimate the size of the population or the number of adults in the population in this time frame. So a little bit of a simplified example here is these 3 individuals, which are sampled at each of these occasions, and each individual is either seen with a 1 or not seen with a 0 at each sampling occasion. So pulling the capture histories of all the individuals, then we can estimate those relevant parameters. And among these relevant parameters is NA, the number of adult individuals in the population. So with this information, we can see how this parameter varies through time. We can obtain some estimates of the number of males and females of each species for every year. Well, not for every year. Some years will be challenging to estimate, to have some estimates. But in the other years, and some years you will have extremely wide confidence intervals that render those estimates useless, basically useless. But with cumulative years of data, you will obtain better and better estimates, and you will account for the variation, for the natural variation in this parameter. Well, notice that you need a high number of marked animals and obtain relatively high recapture rates in order to obtain good estimates. So we published these results in our paper in Ecology and Evolution, and adding more years of data, we can account for more variation in this parameter through time. And moreover, we can we can complement this information on the relative abundance of the adult individuals through years with evidence of breeding activity. For example, we can count the egg strings of this species, Epidalea calamita, through time, and see how this quantification of the breeding success mirrors the fluctuations in abundance. And furthermore, we can try to see the link between these population dynamics and extrinsic factors, such as the rainfall of each year, which turns out to be very important for this species, because the natterjack toad, this Epidalea calamita, happens to breed in extremely shallow pools that are formed after heavy rainfall in the spring. So this is a risky strategy, because tadpoles of this species face a very high risk of massive mortality for desiccation of the pools before they can reach metamorphosis. So most of the years, this strategy ends into breeding failure. But in the years when the rainfall is sufficient for the pools to last enough for the tadpoles to reach metamorphosis, the recruitment of this species is outstanding, as happened in the last year, 2018. So there is an interesting consequence of this breeding strategy, and it is that very fast colonization of these shallow pools after heavy rainfall is critical. If you are going to give any chance for your tadpoles to survive to metamorphosis, you have to colonize those pools as soon as possible. And that, coupled with the mating behavior of this species, which is very long-lasting, more or less 24 hours, a couple is mating, that ends up in a consequence that is that the levels, the polygamy levels in this species is extremely low. So most individuals cannot access to more than one mating couple every year, which ends up in an operational monogamy within each year in this species. So could this mating behavior have something to do with the relative stability in NB that we observe in the 2 years in which we have data? Well, maybe the mating system of this species is related to that. So what is exactly NB? What are we estimating here? So NB, as we said before, is the effective number of breeders in the population. So it's an approximation of the effective size of the population for one single breeding season. And it is not exactly, it cannot be exactly as equalized or quantified as the number of males and females that are breeding. It certainly has to do with it. So if we have the formula here, and this is the number of males and the number of females, but we also have this term S sub k squared. And what is S sub k squared? Well, this is the variance in parental contribution to offspring. So the average breeding success of the individuals. And this is part of the equation, and this is accounted in this parameter. And this is extremely difficult to know what is happening about the parental contribution in natural populations. So what do? Well, we have genetic methods that estimate this quantity. But some assumptions have to be made in order to estimate this quantity, this parameter, with those methods. So what do? We employ one method with our genetic data and just trust the quantity that we obtain. Well, we think that we need something else to calibrate that we are obtaining reliable results. Some information about the mating system of the species is required for those analyses sometimes. So we need to know something about that, but what if we don't know about the basic demography, the natural history of the species, what do we know about this? Well, keep calm. So we have a solution here, and for example, we found the solution in the pedigree analysis. So this is one of the available methods for estimating the effective number of breeders, which is the shift frequency method implemented in program colony. And it is interesting for us because it allows the joint estimation of the NB with the reconstruction of the pedigrees of the samples you have. So if you have, for example, a genetic sample of tadpoles, so a single cohort, so they are from a single cohort, and you may have, it is desirable but not necessary, to have samples of the adults, males and females that you observe in that population. So you have the candidate parents and the candidate mothers for those tadpoles, then you can reconstruct the pedigrees. And for each tadpole you have, you may find that this is the father and this is the mother with an associated probability. And you can do that for all the tadpoles in your sample. And the crucial fact is that this is the method to study the effective size, the NB. But also this gives you some information that you can compare with data that you observe in the field. For example, you can observe mating pairs. You can compare if the mating pairs you observe are consistent with the parentage that you reconstruct based only on genetic data. And you can know how long the individuals spent in the breeding site. So therefore increases the chance of multiple matings. So you can compare the polygamy rates that you obtain with this kind of data. And you can compare the number of egg strings, the evidence of breeding success or breeding activity that you observe in the field with the number of families that you observe in your reconstruct pedigrees. So this is the thing that gives you an idea of how reliable your estimate of NB is. And this is crucial for us. Of course, there are other methods to check or to test that reliability. For example, you could do sensitivity analysis, and you could find that in some species, your estimates for NB are insensitive to the marker information or the different parameters of your analysis. If they turn up stabilizing at some value, then you can be more confident that you are obtaining reliable results. Whereas in other species, maybe as you increase the genetic information, you don't reach such a stabilization. And that means that you probably are not approaching the real value. So you cannot really trust your results in this case. You need more genetic information, or you need some information to compare here to adjust the estimate. So by applying this methodology, we were able to obtain reliable ratios between NB and NA in different species. And we have, with cumulative years of data, you can account for the variation, in the natural variation in this ratio through time or in different populations. And it can give you a lot of information of what are the demographic dynamics intrinsic of the species and what is their role on the evolutionary processes that scale up through time. And even more, if you have information from the field or from your reconstruct pedigrees that tells you which individuals bred successfully, then you can gather information about life history traits of those individuals, for example, the body size or other individual covariates. Then you can try to establish what are the life history traits associated with higher breeding success. So, for example, that was the work of Elena Martinez. It was her graduate thesis work that she presented this very same week. And for example, body size was strongly associated with the reproductive success in the Natterjack toad, in Epidalea calamita. This is not new, of course. Body size has long been associated with the breeding success, but other variables were also associated. For example, the time spent at the breeding site by the individuals, measured as the number of captures that the individual have in the breeding site during the breeding season, was a strong predictor as well of the probability of that individual mating. And not only the probability, but also the number of matings that individual could obtain through the breeding season. So, and turning to the second parameter I wanted to talk to you today, it's about the role of connectivity, the connectivity between different populations. What is the role it's playing in the evolutionary processes? Well, dispersal is a key parameter that we need to account if we want to explain the gene flow that we observed or the genetic structure that we observe in phylogeographic analysis. So we need to know something about how species disperse, and this is quite difficult to obtain as well. We have very little information of the movement ecology of many species, and amphibians are, again, not an exception. So to do that, we normally address classic phylogeographic approaches. For example, we can study one or more species inhabiting the same place in order to test the role of different factors, for example, landscape features, in the genetic structure that we observe in those species. And as an illustration from our own research as well, last year we published this paper in which we evaluated the genetic structure of these 4 species across the central system in Spain. And we observed that the central system is a strong barrier to gene flow for some species, for example, for the sped-foot toad, but it only acts as a semi-permeable barrier for other species like the tree frog, the Iberian tree frog, the natterjack toad, or the Iberian green frog. So, okay, some species show less genetic structure. Some of them show stronger genetic structure. Is that associated with its dispersal capacity? Well, it should. But how can we compare? Indeed, the literature is really scarce in data about the movement, the dispersal capacities of each species. We addressed that question too in our monitoring program, and for that, in the southern slope of the central system, we have our main study area, which is composed by two main ponds. The Laguna de Valdemanco is here, and another artificial pond is here. We call it the mining pond. Here you have these two images are from the Laguna de Valdemanco, which is wider in surface, but is only 1 meter deep. It fills with the winter and summer rains, and it dries up during June. In awful years like this year, is already dried. And also the mining pond is artificial. It has its origin in the mining works that were developed in the last century. And it is smaller in area, but is deeper in surface, so the hydroperiod lasts longer. And here we study a community of 9 species. 7 of them are more common, and I will show you pictures of them. This information accompanying here is the number of individuals that we have marked in these 10 years, the number of males and females, and the rest are juveniles. The recapture rates we obtain, which are quite high because we focus in a relatively small area, which allows us to obtain high recapture rates, but that's what we need to obtain robust or reliable estimates. And this is interesting because it's the maximum lifespan given our data. So we have been here, we have around 10 years of data. We are marking adult individuals, so based on the time to maturation, we can obtain a minimum estimate of the maximum lifespan of each species, which is not complete, of course. This species could live much longer, but simply our study hasn't lasted so much by now. But we have an estimate at least, so and we can compare. So that was the sharp-ribbed newt, the Pleurodeles walt, and we also have the marble newt, this beautiful newt here, Triturus marmoratus. And among the anurans, we have the midweight toad, which has this amazing behavior of the male carrying the egg clutches for some weeks before releasing them in the water. We have the sped-foot toad. We have the natterjack toad, and the Iberian tree frog, Ila moyeri, and the green frog, Pelophylax perezi. And then if we want, we mark them with passive integrated transponders. If we wanted to obtain good data of the movements of the individuals, we will need a different approach. We will need radio tracking or something like that. We don't have that. So we don't have the resources to employ large sizes, expensive devices for as many individuals as we would like, of course, but since we are marking them with passive devices, we can record that we mark it in some place and we recapture in some other place. So we have an estimate of the movement. So for example, this is one individual that we mark in this date in the Laguna de Valdemanco, and we recaptured 4 years later in the mining pond. And then 1 month from then, we recaptured then again in the Laguna de Valdemanco. So of course, this is a minimum estimate. Of course, the movement of this individual probably was much higher than this, but we at least have some estimate. Of course, this is dependent on our sampling scheme of the places we search, and of course, the longer the distance, the real distance that individuals move, the lower the chances of us recapturing that individual and accounting and recording that distance. But with all these years and all these individuals marked and these high recapture rates, we think that we can have some idea of the movement of each species. And for example, for Pelophylax perezi, in all these years and with 900 individuals marked, we observed 9 displacements from the Laguna de Valdemanco to the mining pond and 12 the other way around. So it's not high numbers, indeed. But we also observe movements to other minor breeding sites like water trucks, abandoned quarries, or an abandoned swimming pool, or a stream that we have here. And also travels back to the same places of marking, and also movements to the mining pond and the stream. And so we have some idea of how this species is moving through the landscape. And we can compare this with other species because we visit the same places surveying all these species. And what do we find? Well, we find that not all species move as much as green frogs do. The ribbon newt, for example, with a lot of individuals marked and the same monitoring program, show very low levels of very low number of frequency of movements between those places. So this species is moving less than the other species. We have relative proportions of the movements, but indeed it does. So when we check at the genetic information, and that was the master thesis of Iñaki Fernández de la Rea, we observe if we obtain genetic samples of the Laguna de Valdemanco and the mining pond, we observe a strongly asymmetric gene flow between these two ponds. And why is that? If this animal is moving so little, why do we observe such an asymmetric pattern? Indeed, he found that a large proportion of the individual samples here were not directly migrants from the Laguna de Valdemanco, but second generation migrants, so grandchildren of individuals that migrated from the Laguna de Valdemanco. Okay, again, we obtain some inference from genetic information. Can we calibrate? Can we trust this? Can we calibrate it somehow? Well, in this case, we are talking about an artificial pond. It didn't exist. If we check ancient photographs, it didn't exist some decades ago. So we can look at to the ancient photographs and check when pools started to appear in this place. And it turns out that in the 1970s, 1980s, the first pools started to appear here. So before, there was not population here of this species. The only population could be here in Laguna de Valdemanco. But starting in this date, probably a colonization could start. And this is consistent with the pattern we observe. The times are consistent with that, ending up as the two populations that we have today, that even though they exchange a low number of individuals nowadays, it turns out that this population was founded by individuals from this population. So to summarize the take-home messages I wanted to express today. So natural history and demography are very important. We need a solid background in these things about the species in order to explain the evolutionary inferences that we make from the patterns we observed. And we are confident, we think our results, our work supports that an integrative approach combining different lines of evidence, but mainly genetic and field-based capture-mark-recapture work, is able to produce reliable, robust, reliable, and comparable estimates of different demographic parameters. From these relevant demographic parameters, the effective to census size ratio is very, very important to obtain some insights of the natural population dynamics and how intrinsic life history traits or extrinsic, for example, rainfall or environmental stochasticity, drive the dynamics that we observe in the population. And talking about the connectivity between populations, this is variable within a species, like in this example we saw of the sharp-ribbed newt. So it can change through time. The dispersal rate can change through time, depending on also, again, in intrinsic and extrinsic factors of the population and extrinsic factors of the environment. And also, connectivity is different between different species inhabiting the same place. So this gives us insights about the role of different barriers to the gene flow through time, and this has to be consistent with the records of dispersal capacity that we observe. If we get that, then we can predict that genetic structure is predictable. And that is all I wanted to say today. I want to thank the funding, the members of the group, and thank you for your attention.